botulus is sausage. It is Latin word. First, it was identified from the person who has ingested sausage. So, according to Latin word botulus, the name of disease is called botulism. Causative agent of botulism, already I said, is bacteria named botulism. If we observe morphology of Clostridium botulinum, it is gram-positive bacilli. It is gram-positive bacilli. It is motile. It is spore-bearing. It has subterminal and oval spore. It is gram-positive bacilli. It is motile. It is spore-bearing. Shape and position of spore is subterminal and oval, but it is non-capsulated. So in morphology of Clostridium botulinum, it is gram-positive bacilli, motile, spore-bearing, and non-capsulated. It has subterminal and oval spore. This bacteria, it is anaerobic bacteria. It is obligate anaerobic. It strictly requires absence of oxygen for its growth. It is obligate anaerobe. It grows on blood agar, nutrient agar, anaerobically, or in anaerobic culture media such as RCM, Robertson's cooked meat media. It can grow at 35 degrees Celsius. So in cultural characters, for Clostridium botulinum, it is obligate anaerobe. It can grow in blood agar and nutrient agar anaerobically, or better it grows in anaerobic culture media, Robertson's cooked meat media at 35 degrees Celsius. There are 8 types of Clostridium botulinum on the basis of antigenicity. On the basis of antigenicity, Clostridium botulinum, it is categorized into 8 types. Type A, B, C1, C2, D, E, F, and G. Among these 8 types of Clostridium botulinum, type A, B, E, and F cause human botulism. On the basis of antigenicity, Clostridium botulinum is categorized into types. Clostridium botulinum, type A, type B, type E, and type F, they cause human botulism. Type A, B, and F are proteolytic. Proteolytic means if we grow type A, B, and F, Clostridium botulinum on RCM, Robertson's cooked meat media, they will produce black color. Proteolytic C, D, and E are saccharolytic. Saccharolytic means they will produce a red color on RCM, Robertson's cooked meat media. Type C and D, they do not cause human botulism. So among type A, B, E, and F, which cause human botulism, they may be either proteolytic or saccharolytic. Type A, B, and F are proteolytic. Type E is saccharolytic. Now, pathogenesis. How Clostridium botulinum it can cause infection? Source of infection is undercooked sausage, or wounds, or soil. Mode of transmission, mostly it is by ingestion. And mechanism, strains of Clostridium botulinum type A, B, E, and F, they produce neurotoxin. Type A, B, E, and F Clostridium botulinum, which cause human botulism. First, how they will lead to botulism? First, they will produce neurotoxin. After ingestion of Clostridium botulinum, toxin is liberated during growth and during autolysis in gut. Botulinum toxin absorbed from gut and binds to receptors of presynaptic membrane of motor neurons of peripheral nervous system and cranial nerves. Botulinum toxin, it is absorbed from gut and binds to receptors of presynaptic membrane of motor neurons of peripheral nervous system and cranial nerves. It will lead to inhibition of release of neurotransmitter, mostly acetylcholine. It will lead to inhibition of release of neurotransmitter acetylcholine at the synapse, which will lead to lack of muscle contraction and symmetric descending paralysis. Death in botulism is due to respiratory paralysis. This is general mechanism of pathogenesis. But there are three clinical types of botulism: foodborne botulism, wound botulism, and infant botulism. So, mechanism slightly differs in different clinical types of botulism. There are three clinical types of botulism: foodborne botulism, wound botulism, and infant botulism. Foodborne botulism, it is paralytic disease caused by ingestion of preformed toxin in food contaminated with Clostridium botulinum. Foodborne botulism, it is paralytic disease caused by ingestion of preformed toxin in food contaminated with Clostridium botulinum. Incubation period for foodborne botulism, it ranges from 12 to 36 hours. Remember, hours. Incubation period ranges from 12 to 36 hours. Symptoms of foodborne botulism includes vomiting, thirst, constipation, visual disturbance, dysphagia, difficulty in swallowing, difficulty in speaking and breathing. Foodborne botulism, mostly it is due to ingestion of contaminated sources. Incubation period, it ranges from 12 to 36 hours. Symptoms include vomiting, thirst, constipation, visual disturbance, dysphagia, difficulty in speaking and breathing. It will lead to flaccid paralysis. Due to paralysis of cranial nerves, there will be weakness of peripheral muscles. There will be weakness of peripheral muscles. There will be urinary retention and respiratory paralysis and ultimately death. Due to respiratory paralysis, death may occur in foodborne botulism. Other clinical type of botulism is infant botulism. There are three clinical types of botulism: foodborne botulism, infant botulism, and wound botulism. First, foodborne botulism, that is due to ingestion of sauces, contaminated sauces. Next is infant botulism. Infant botulism, it occurs due to feeding of honey. Remember, your MCQ ma sodhna sakcha. Infant botulism, it is due to feeding of honey contaminated with spores of Clostridium botulinum. Infant botulism occurs due to feeding of honey contaminated with spores of Clostridium botulinum. Spores germinate in gut, multiply, and produce toxin. It is seen in infants below 6 months age. Infant botulism, it is seen in infants below 6 months age due to feeding of honey contaminated with the spores of Clostridium botulinum, but it is not seen in older children and adults. Features of infant botulism, they are poor feeding. Clinical features of infant botulism, it includes poor feeding, weakness, and signs of paralysis. There will be poor feeding, weakness and signs of paralysis. constipation, there will be constipation, there will be pooled oral secretions. Pooled भनेको यो थुक्को फिजहरु मुखको oral secretionहरु pooled भनेको जम्मा हुने. Pooled oral secretions, and there will be altered cry. So features of infant botulism includes poor feeding, weakness, and signs of paralysis, constipation, pooled oral secretions, and altered cry. Other clinical type of botulism is wound botulism. First is foodborne, second is infant, third is wound botulism. It results from wound infection with Clostridium botulinum. Wound botulism, it results from wound infection with Clostridium botulinum. Toxin produced by Clostridium botulinum at the site of wound infection is absorbed in blood. Toxin produced by Clostridium botulinum at the site of wound infection is absorbed in blood, and neurological manifestations occur, except manifestations such as difficulty in swallowing, speaking, breathing, delirium, coma, and death. Wound botulism, there will be, it occurs due to absorption of toxin in blood through wound where Clostridium botulinum releases toxin, and neurological manifestations occur except manifestations such as difficulty in swallowing, speaking, breathing, delirium, coma, and death. So these are three clinical types of botulism: foodborne, wound, and infant botulism. Now, how will perform lab diagnosis of botulism? Lab diagnosis of botulism, diagnosis is done mainly clinically by observing clinical features, but for lab diagnosis, for confirmatory diagnosis, we have to perform lab diagnosis. Lab diagnosis, it can be done by demonstration of toxin. It can be done by two methods. Lab diagnosis of botulism, it can be done by two methods. First, demonstration of toxin, other demonstration of bacteria in suspected residual food, patient's serum, intestinal contents, feces, etc. So one is demonstration of toxin, other is demonstration of bacteria. First, demonstration of bacteria. It can be done by gram staining. Clostridium botulinum, it is gram-positive spore-bearing bacilli. It is gram-positive bacilli which contains spore. Spores are oval and subterminal. By observing this morphology in gram staining, we can identify Clostridium botulinum. Other, we can perform culture of Clostridium botulinum. It can be done in Robertson's cooked meat media, RCM broth that you have seen. It can be done in Robertson's cooked meat media. Type A, B, and F. Type A, B, E, and F. These four types of Clostridium botulinum, they cause human botulism. Type A, B, and F, they are proteolytic. E is saccharolytic. So any type of the Clostridium botulinum which causes human botulism, from clinical sample of botulism, if we perform culture in RCM, Robertson's cooked meat media, we may get either a proteolytic reaction, turning of RCM media into black color, or saccharolytic reaction, turning of RCM media into red color. RCM media may turn into black color, that is proteolytic reaction. RCM media may turn into red color, that is saccharolytic reaction. we may get any type of reaction in case of botulism. Other method for lab diagnosis of botulism is demonstration of toxin. First, we have to take sample, that residual food, vomit, whatever it may be. That sample is mixed in sterile isotonic saline, macerated, mixed, and filtered. Is divided into three parts. That filtered extract sample is divided into three parts. One part is heated and two parts are unheated. Remember, we have taken sample mixed in sterile isotonic saline, macerated and filtered. That filtered extract is sample is divided into three parts. One part is heated at 100 degrees Celsius for 10 minutes. One part is heated and two parts are unheated. Then two mice are injected. We have to take two mice. Total we have to take three mice. Two among which two mice are injected intraperitoneally. Remember, we have to inject intraperitoneally. Two mice are injected in which is protected with antitoxin. Third animal but active in risky group people only. First dose 2 ml is given in first dose. After 10 weeks, second dose is given, and after 1 year, booster dose is given. This is about active and passive immunization. Clinical application of botulinum toxin. Botulinum toxin, it is used. Botulinum toxin, it is responsible for botulism, but it has clinical application also. Botulinum toxin, it is used for treatment of strabismus. Botulinum toxin, it can be used for treatment of strabismus. This is about botulism. Botulism, it is paralytic disease due to neurotoxin produced by Clostridium botulinum. There are 8 types of Clostridium botulinum, among which 4 types of Clostridium botulinum cause human botulism. Type A, B, E, and F, they cause human botulism. Type A, B, and F, they are proteolytic. Type E, it is saccharolytic. Clostridium botulinum, it is obligate anaerobic bacteria, so it is grown on mostly Robertson's cooked meat media, which is anaerobic media. There are 3 clinical types of botulism: foodborne botulism, wound botulism, and infant botulism. Foodborne botulism, it is due to ingestion of mostly sauces contaminated with Clostridium botulinum. Bacteria, it releases botulinum toxin. This botulinum toxin is responsible for disease botulism. In case of infant botulism, mostly among children, infants below 6 months age, due to ingestion of honey contaminated with Clostridium botulinum, it leads to infant botulism. In case of wounds, if there is presence of Clostridium botulinum, if it releases botulinum toxin due to absorption of botulinum toxin in blood, it will lead to clinical features of wound botulism. There are three clinical types of botulism: foodborne, wound, and infant botulism. Mostly diagnosis of botulism is done clinically, but for confirmation, lab diagnosis should be done. Lab diagnosis, it can be done in two ways. One is demonstration of bacteria. It is gram-positive bacilli having spore. In gram staining, gram-positive spore-bearing bacilli. Demonstration of gram-positive spore-bearing bacilli confirms it is Clostridium botulinum. Similarly, we can perform toxin demonstration. Toxin demonstration, we have to take sample. Filtered extract is divided into three parts. One is heated, two parts are unheated, and 3 mice are injected intraperitoneally. And after giving injection intraperitoneally to 3 mice, mice in symptoms. Among 2 mice in which we have given unheated toxin, one mice is protected with one mice is given antitoxin, and in result that antitoxin provided mice, there will be no appearance of symptoms. But in mice in which we have given unheated filtered extract without antitoxin, we have not given antitoxin, there will be appearance of symptoms, flaccid paralysis, and death of mice. In this way, we can demonstrate, we can perform lab diagnosis of botulism by demonstration of toxin also. Any query? Any query? Now I will take attendance, just wait, okay?